that there's an external day and night cycle, there's an internal circadian clock that is separate from the external day and night cycle. However, using the theory of oscillators, what we learned is that these two oscillators get entrained, and by that, what we mean is that even though these curves, this red, green ones, did not originally have the same frequency as the day-night cycle, after coupling and entrainment, you have them oscillating with the same time period, which is 24 hours. However, the second important thing we learned is that because of this idea of the phase of entrainment, which we derived yesterday, what will happen is that these oscillators will be phase shifted, right? So you can clearly see that when your day-night cycle is here, for example, let's say this afternoon, one person's oscillator might be here, the other person's oscillator might be here. So the times that each of these individuals' bodies are reading out is somewhat different. Okay, and this was the idea that's called chronotype, and the physics behind this is simply that of the phase of entry. Okay, all right. So that was what we covered yesterday. Today, what we are going to discuss is, so remember, this was all quite phenomenological, right? This was in the spirit of classic physics modeling, where we really did not care about the molecular details of these observations. But next, the question that arose in the field, this is say around 1960s, 1970s, is when we understand the basic physics of these oscillators, but what exactly is setting up these oscillations in the human body? Okay, what are these things that we can actually measure? So this is going to be the main focus of today's talk. Is what creates these oscillations in us, and once we can figure out what creates these oscillations, how do we make measurements? And then once we have data from these measurements, the big question that we're going to try to answer today is how do we actually infer the individual's body type? Okay, so that's a very, very exciting question right now. This is the present. In the last 10 years, the field has been trying to answer. Okay, so that's largely what we're going to do. And today, the kind of physics that I'm going to introduce to you is linear algebra. And the hope is that it's not going to be too much more than what you already studied, but what you learn from today's lecture is a very, very popular and helpful tool that people in the linear algebra machine learning field use. And I'll show you how that is used to infer chronotypes or body time from real data sets. Okay? All right. So, of course, if you remember the sort of timelines we were talking about, we ended up in the 1950s when people started making these connections between these real world oscillators and things like entrainment. Right? So then the next question was, how are these coming up in mammalian systems or in organisms? And the first classic example that came to people's mind were these very, very sort of groundbreaking experiments that this Russian chemist Boris Belousov had done in 1951. Okay, so at that time, historically, people did not believe that chemical reactions can show oscillations. So people thought that equilibrium in these chemical reactions will necessarily be obtained in a monotonic fashion. Okay, so there cannot be any non-monotonicity in chemical reactions as they approach equilibrium. So his experiments were very interesting. He was working in the lab. He took a bunch of chemicals, and what he noticed was when he mixed them up, he saw that the color changed and oscillated. So first it started off with a certain color. I don't think these are exactly the colors he originally saw. This is a replication of the Belousov experiment. But he noticed that the color changed, for example, here from brown to blue, back to brown to blue, and lasted on the order of hours. Okay, so for 6 years he couldn't publish these results because no one believed him. Everyone said what you're doing is complete nonsense because this simply cannot happen in chemical reactions. So it took him 6 years, and after which he published this in some godforsaken journal. But this is a good lesson for all of you, right? There were questions being asked about how do you know whether there's a mistake in what you're doing or it's a real experiment. You have to stick to these things. Sometimes when you find really new phenomena, you find it very hard to actually publish this. But anyway, so after that, what happened was a graduate student in someone else's lab, Zabotinsky, was given these experiments and he managed to replicate this over and over again. Okay? And ultimately, it was with him that they published these results. And then it turns out that a lot of theoretical physicists jumped onto this problem to try and understand how do chemical reactions operate. And so there's a long history, which I will not go into if you're interested. One of the first models of the Belousov-Zamotinsky type reaction was provided by Goodwin. It was an extremely controversial paper. For almost 20 years, people disputed even these theoretical results. They said that Goodwin had made mistakes, and that's why he had found oscillations, etc. It's a very interesting history. If you want to read it, go ahead and read this. Prigozhin, who won a Nobel Prize in chemistry for doing things in non-equilibrium thermodynamics, also worked on this problem of trying to understand when do chemical reactions oscillate. Okay? And ultimately, people in the context of circadian clock model the circadian clock, and two famous people here are Brinkley and Kuramoto. Some of you may have heard of Kuramoto. He's famous for his coupled oscillator system. Right? Okay, so I will not go into the details of these models because these are much more nonlinear dynamics, dynamical systems kind of approaches, which we touched on yesterday. And I want to do something different today. But I just want to leave you with the intuition behind why chemical reactions might oscillate. Okay? And what I want to do is give you that intuition with an analogy to a bathroom shower. Okay? Because this is something I'm sure all of you have experienced, right? Especially in Delhi winters when you wake up, it's freezing cold. You go to the shower. Of course, you want your water to be hot. So you yank the knob to the left, right? And then, of course, what has happened is there's a delay, right? The system doesn't generate the right temperature immediately. So you get your skin gets burnt, right? Immediately. So then what is your reaction? You react it back to cold, and then you're suddenly shivering. Right? So notice two things that's happening. So ultimately, what ends up happening is that you start too hot, then the temperature becomes too cold, then it goes to slightly hot you want. And so essentially what you have set up is a oscillation. Okay? That's what I'm trying to get at. So the two very important things in this analogy is, remember I told you that there has to be a delay, right? There has to be a delay in you turning the knob and system detecting the temperature. One very important thing. And the second important thing, notice is there is a negative feedback in this system. When it becomes too hot, your body tells you that this is too hot, I need to reduce the temperature. Then when it becomes too cold, your body tells you you have to increase the temperature. So this is classically what is known as a negative feedback system. Okay? So you see two important things that allow you to generate oscillations. That's a negative feedback and a delay. Okay? And it turns out that it's a little bit more mathematically involved, so we may not go into this, is that you also need nonlinearity in your equations. Okay? I'll show you how this comes. So these three concepts became very popular during the 1970s and 80s, where all these physicists working together gave the intuition that these kinds of things need to be expected to exist in your chemical reaction system so that you see sustained oscillations. And so, of course, around that time, the 1970s, 1980s, was the time of the molecular biology revolution. So this was the time when you started being able to manipulate genes at will. You were inserting genes, you were taking them out, you were cloning them, you were sequencing them, you were basically doing all kinds of things to biological systems. And what happened was, because of the work of a lot of people, including these three who I've shown here, this is Michael Rosbash, Jeffrey Hall, and Michael Young, all working on flies and trying to figure out the underlying chemical oscillations that give rise to the circadian clock. These guys, of course, won the Nobel Prize in medicine in 2017 for figuring out exactly what are these reactions that give rise to the circadian clock that we've been studying. Okay. Of course, there are other people involved as well. Seymour Windsor, Konopka, some of these famous people who are the founders of molecular biology were also involved in this thing. So ultimately, what came out, and this was consistent with these concepts that came out from dynamical systems theory, is a simple picture of how the circadian clock oscillations arise in mammalian systems. Okay, so what happens is that, forget the names, these names don't matter much, but what you need to know is that this black line represents DNA. How many of you have heard of the term called genes? Wow, I had no idea what a gene was when I was in physics. Something has changed, right? Okay, so basically, just if anyone doesn't know, a gene is a small stretch of DNA which gives rise to a certain protein. Okay, so that's going to be our working definition of a gene. So you have a small piece of DNA. Forget what this name is, right? This DNA gives rise to an mRNA, which is called gene expression. We'll be using that term quite a lot today. Gene expression means DNA giving rise to some mRNA molecules. That's called gene expression. And then that mRNA gets converted into protein. Okay, that's all you need to know. So what happens in the circadian clock system is that these two proteins come and switch on the production of these mRNAs. Okay, so think of this as a switch. When it comes and binds over here, it's part of the DNA. Your mRNA starts getting produced. The mRNA has to be shuttled into the cytoplasm. mRNA is created in the nucleus. It takes a little bit of time to get shuttled into the cytoplasm. So this is your delay. Okay, remember I was talking about the delay from the last slide. So that bit is your delay. And then these guys, they come back, shuttle back into the nucleus and bind to these proteins and prevent them from binding here. Okay, so this is my negative feedback system. You can see that what was creating these mRNA in the first place are themselves going back and switching off the production of that same mRNA. Okay, so this is a negative feedback system, and you have the delay. So you have the two main components that gives rise to oscillations. There is also some non-linearity involved here because you have heterodimers, meaning two different molecules coming binding together to create this switch-like behavior. You can show that this creates non-linearity, so I'm not going to go into that. But this is sufficient to create what we have been discussing, these oscillations of the circadian clock. Okay, so yesterday what we were sort of talking about, these oscillations, you can think of these as arising from chemical reactions, and the y-axis is just the number of mRNA molecules or protein molecules. Okay, so that's what is meant by a circadian clock oscillation. These are, of course, roughly of the time period of 24 hours. So you can see these are real data sets over here. So you can see that these individual colors correspond to these species. So these are all, you can think of as chemicals, the proteins, mRNA, and so on. Their numbers are being plotted on the y-axis. This is time, and you can see how you can find these beautiful oscillations of these species that come out of these genetic networks. Okay. Any questions on this? So this is the real thing that is going on that we were trying to model yesterday. Okay, and this is extremely important because based on this knowledge, we are going to create experiments that allow us to generate data. Okay? So I will give you one example from our lab. So this is in fact a very close collaborator of mine, Adrian Granada, who's at the Charité. These are the kinds of experiments that we are also doing in the lab. So what you're seeing here are single cells. Okay, so this should fascinate you. Each of these things are a single cell. These single cells, we've engineered them to light up. So what we've done is we've tagged one of these proteins. So in kinds of light on it. So that's the color that you're seeing here. So focus on, for example, this cell. What you'll notice is that the intensity of this color rises and falls, okay, which suggests that there is a circadian oscillation. So these are what are called time-lapse microscopy visuals. So we take these cells in the lab, we put a microscope on them with something like 20x or 30x magnification. These are called fluorescence microscopy experiments where there's a light that shines which allows you to see this fluorophore, and then you can create time series out of it. So just see this movie once, you'll see what real experiments look like. Just stare at the intensity over here. You can see that it went down, and as the cell is dividing, you can see that the intensity is going down and coming back up. And of course, there's a lot of cell division going on. This is because we are working with a particular cancer cell line. Once you take that intensity, you run it through certain algorithms. These are, again, very sophisticated machine learning algorithms that detect cells from your field of view. You can extract these intensity profiles. So this was done by a student of mine. You can see that these oscillations are approximately 24 hours. Okay. What you will also notice, and this is where really the fun of complex biology comes, biology does not look like this, what you study in physics, okay? When we do physics, when we do simplified models, you assume that you have things that are beautiful sine waves, right? This is not true in reality. What you will see in reality are oscillations that look like this. This is way noisier. You can also see that even within a single trajectory, the time period in fact changes. So all of these add complexity to your biological problem, but this is the sort of fun in biology that things are so noisy. Anyway, so you get a sense of what is now creating these oscillations. So these are all chemical reactions happening. So then now, just touching base to what we were talking about yesterday, once again, we are thinking about chronotypes, and now we have to think about chronotypes in the context of genes and proteins. Okay, so each individual, so let's say we are talking about two individuals, they have different body types. The reason is because you have these genes that are oscillating. Okay, genes have specific phase differences. That is very important, right? These network architectures that you have here give rise to genes that oscillate with certain phase differences, fixed phase differences. Because remember, when this guy is very high, it starts creating this guy. Then these per and tri molecules take some time to come back and then switch these off, right? So there are fixed phase differences. So when one thing starts going high, after a certain amount of time, the others are going to go high, and that's why your curves look like this. Okay, some are peaking, some are at the trough, and so on. Okay, this is very important because this will be of fundamental importance in all our machine learning algorithms later. Okay, so and then what you can do is you can arbitrarily define one of the genes, the peak, to give phase 0. Okay, and the rest of the genes have fixed phase differences to your gene of whatever, the one that you've used to define what is the phase. So then roughly the idea is that these gene expression patterns and their relationship to the time of the outside determines what is meant by body type. Okay, so this has now got translated into a real picture where your oscillations are genes, and the relationship to the oscillation of these genes to the time of the outside sets what is called body type. Okay, any questions about this? This should be clear enough. Okay, so in the last 10 years, what has become incredibly exciting is the understanding that this body time of ours controls hundreds of different things, physiological processes in the body. Okay, so for example, in cancer cells which are dividing, these oscillations, time of your body controls how fast cancer cells are proliferating. For example, right? It controls aging processes. It controls your microbiome, the bacteria that is in your gut. That is strongly coupled to these oscillations. Okay, so if you measure your gut microbiome, species diversity changes as a function of day. Your immune response, right? It has recently been shown over and over again that the number of T cells in your body that roam around, that are doing all the fighting against bacteria, are directly connected to these oscillations. Okay? So figuring out what an individual's body time is has become one of the most exciting questions in chronobiology in the last 10 years. Okay? And as you can see here, this is sort of the kind of thing that people are trying to do in the present moment, where you have a sample of a person, or let's say from many people. So this is sort of the standard thing that is currently being done. You take a blood sample, for example, you isolate all the cells from the sample, so you have millions and millions of cells. You grind them up and you measure these genes. Remember, when I say you measure these genes, it means that you're measuring the amount of mRNA that exists in that sample. Okay? So you measure the gene expression of these samples, and then you come up with clever algorithms that allow you to infer this body time of an individual. Okay? And by body time, remember what I mean once again is the relationship of these gene oscillations to the external daylight cycle. Okay? So this, in the next half an hour, what I'm going to do is I'm going to switch over to the board and I'm going to show you one such algorithm, which is, I think, a very clever algorithm that allows you to do this inference from data sets, these modern data sets from these kind of sequencing steps. Okay, so that's what I'm going to do in the next half an hour, and this will hopefully be sort of familiar to you because you probably studied a little bit of linear algebra. So what I'm going to show you is how a simple advance of the kind of linear algebra that you've done will allow you to answer questions like how you extract body types. Okay? All right. So what we do is we move to the board. I'll still keep this up for a little bit. So what I want to first do is formalize the problem a little bit. I've sort of given you the background of what we want to do and why we want to do this. But let me formally define the problem a little bit so you can start thinking about it mathematically. Okay, so this will not work on the board. Okay, so let's say you want to formulate the problem. Formulate the time inference problem. So, what do experimental measurements look like? Okay, so what typically we get out of modern biology, these are methods that are based on sequencing. I won't go into too much detail, but what you land up getting are matrices that look like this. So we call the data matrix X. You can still hear me, right? Okay. So throughout the lecture, my matrices will be denoted with a tilde. Okay, vectors will have a dash. So what data looks like is the following. On the rows, you have genes, and here on the columns, you have samples. And I'll tell you what this means. So, for example, we could have G1, G2, and so on, all the way up to D genes. Okay, so we are thinking of a three-dimensional space with G1, G2, and so on representing genes. And typically, with the modern experiments that we have, this D can be of the order of 10,000. Okay, you can measure 10,000 genes in any given sample. And here you have columns by that? Remember this picture? So you go into the clinic, you give a blood sample, right? And from that, you make measurements of the gene expression. So samples are from different tissues of your body, or for example, from different individuals. Okay? What you're doing here, each entry, for example, each entry will be a number, and that number represents how many mRNA molecules of the gene G2 existed in the sample S1. Okay? Sample S1 has millions of cells. This is an interesting point to keep in mind, because tomorrow what we are going to try and show is that what kind of new physics do you need if you want to push this to a single cell? Okay, so we are not thinking about single cells right now. S1 has a million cells. G2 is the average gene expression from a million cells. Okay? So that's what my data matrix is going to look like. So the entries here, just to make sure that remember this, entries are gene expression balance. Expression balance. This is a particular history book. This is so that's the thing, right? We don't know what body time we've taken the samples from, right? So if I now take it from all of us, each one will have a different body time, right? So S1, S2, Sn, up to Sn will represent different body types. And that is what we are trying to figure out. We are now going to try to figure out how to order this on a circle. Okay? So the question is, given this kind of a matrix X, can we order S1, S2 all the way up to Sn on a circle? Okay, why a circle? That's obvious, right? We are thinking of a phase variable here that runs from 0 to 2 pi. Okay, so what we want to do is say that, say my sample S1 is phase 0, so sample S1 is over here, right? This phase is 0. Say sample S2 is somewhere over here. This is the phase angle corresponds to S2. Maybe S3 is over here, right? Maybe S4 is over here. This is the order that we are trying to predict. Okay? Small note, if your oscillations were completely clean and noiseless, this is a trivial problem, right? Because if you remember, because of that network architecture, you have a very fixed relationship between each gene. Okay? The blue gene will always be at its peak when this dark blue gene is at its trough. Okay? So if your oscillations were pure sine waves with no noise, nothing, you could just read off these numbers, right? And that would immediately tell you where you were in this oscillation, right? You just have to measure these numbers, and the ratio of these numbers will tell you where in the oscillation you were, because these gene expression values give you a unique phase if everything was noiseless. Yeah, sorry, you had a question.
I'm just trying to clarify.
Yeah.
By gene you mean protein of protein, right?
Correct.
These are, when you are calling them G1 to G0, each of them are unique.
Each of them are different genes.
And these oscillations are unique to every gene.
These oscillations are unique in the sense that they are oscillating with these kinds of patterns, but they have fixed relationships with each other.
And you are going to figure out from the phase.
But no, we know the kinds of relationships that each gene has with each other simply from this network. If this is connected to this via this network, then these genes will peak before these, right? Because these guys are creating these proteins. So this must come before this per-cry proteins. Right? So then B1 and clock, these two proteins and their gene expression will peak slightly before these guys peak.
In the clones,
Yeah.
when you are drawing these oscillations,
Right.
does it match for, like, what are the possibilities of two
I'm not
I know that that's a correct question to ask.
Yeah, I'm not sure I understand the question completely, but the point is that I'll just maybe try to explain what you're saying. So these genes will oscillate with fixed phase relationship with each other. Okay? Whenever the green gene is at its peak, blue will be at its trough. What is chronotype is when this green gene is peaking. Is it peaking at 12 p.m. like this guy? Or is it peaking at 9 a.m. like in this guy? That is chronotype.
So my question is
Yeah.
that when you had gene 1 and gene 2,
Yeah.
you see that one of them is actually here,
Right.
and vice versa. So when I have millions of genes here, or whatever, 10,000 genes here, so do I see these oscillations exactly different unique for each one of them, or there can be nothing? And then actually,
Yeah, maybe we can take this question off. I'm not sure what you mean by unique oscillation. Yeah. So maybe we can chat after this. Yeah, okay. I can already see that we're going to run super late. But we'll see what we can do about this. Okay. So the question here to everyone. Yeah. So we have these samples, we want to assign them phase values. And remember, these are super noisy oscillations, the way I showed you in the real data. Okay? And that's the reason why you simply can't beat off these gene expression numbers. And you need some sort of machine learning algorithm to detect this. I hope this is okay. So it turns out that the key insight to solving this problem, this is one way of doing it. There are many ways people have come up with. One way of doing this is to notice that in high dimensional space, if you look at the gene expression as your dimension, so G1 to GB, if they form your d-dimensional space, then your gene expression of the circadian clock genes will look like an ellipse. Okay, so this is the key insight. Let me show you a code that I wrote that will demonstrate this. Okay. And as I mentioned, you install in R. Very strong advocate of R if you want to use statistical inference. Okay, so what I've done here is I basically just created 3 genes. Let me just plot this out for you. So this is an example of a noisy gene oscillation. Okay, this is what we've been talking about. So let's call this G1, the first gene. And I've done the same thing. I've generated oscillations for 3 different genes. You can see that over here. Right? All I've done is these are sine oscillations, pure sine oscillations, but I've added some noise to it. Okay, so I have 3 genes with some phase shifts. So this one is 1.5, this is 3. So you have these 3 genes. Okay, I can plot the other genes also for you just to show what they look like. So this is the other gene, and then if I plot G3, you can see that they all have phase differences. Okay. Now let me plot G1 comma G2. Okay. What this means is I'm just my axes are gene expression. Okay. So look at this. Plot G1 comma G2. See what it looks like. Yeah. In 2 dimensions, it looks like an ellipse. So if you just think about it, it's actually very obvious to understand why. Because you have these oscillations with fixed phase differences. And if I now plot this in 3 dimensions, notice what it's looking like. In 3 dimensions, you can see a beautiful ellipse. Yeah, can you see that? And also notice I color coded this. So samples that are coming from similar phases are in similar area regions. Okay, the color denotes the phase of each sample. So you can see all the yellows are here, the purples are here, and so on. Okay. So this is a key observation people made. So the question is then in high dimensional space, notice this is just 3 dimensions, so it's very easy to visualize this. But your data here, you have 10,000 genes. So you have a high dimensional space with 10,000 dimensions. You have to identify ellipses like that in this high dimension, right? You can obviously not visualize things. Yeah. So how do you identify this? That's the main question. So many people came up with many methods. There are naive methods of doing this, where, for example, the simplest thing that you can think of is you take pairwise genes, just the way I showed you over here, right? If you plot genes pairwise, you expect to see ellipses in 2 dimensions. Right? So people tried this. They said, okay, let's take genes pairwise, fit ellipses to the data sets. But you can imagine this is a complete nightmare, right? Because you have 10,000 genes, you have to take pairwise genes, and you have to fit ellipses to the data. Essentially, this is a never-ending problem, right? It's a terrible solution to this problem. So the solution that is quite clever, and it works sometimes, again, it doesn't work all the time, is to realize that if you see the variation in the data, you see that it's clearly maximum along this major axis of the data set. Yeah. And the second axis gives you the second largest amount of variation. So this is the insight that we're going to use to try and identify ellipses from high dimensional data sets. The question then becomes, can we identify this direction? Okay? Because if we identify this direction over here, then it's easy. So now let's say a human being comes into the clinic. You take a sample from this person. You can just project that sample onto that direction and the second direction. So if those data points are close to these data points, remember a projection means that if you have this direction, you project data onto that axis, then you will be able to figure out whether those points are close to these points or not. Right? So you will know that the phase of that new sample is somewhere over here as opposed to here. Okay, if you're projecting onto this direction as well as onto this direction. Yeah,
This essentially is what okay, when I say it's just a two-dimensional manifold.
it's a
You
two-dimensional.
just try to orient the coordinate system so that it lies in one of the
Yes,
spaces.
exactly. Yeah.
Maybe that's intuitively easier to understand.
Yeah, so that's exactly what I'm trying to say. So ultimately what Dr. Gupta is saying is exactly what we are trying to do. We are ultimately in this high-dimensional space, we are going to try to do a coordinate transform in a way that we ultimately get this axis. And this essentially is called, it goes by the name of principal component analysis. Okay? Principal component analysis or PCA. So this is sort of your machine learning 101. And we'll just quickly go through the intuition exactly like what Mr. Dr. Gupta was saying, what the intuition is behind finding these directions. Okay? All right. So I'm going to remove this now because I need the board. So once again, is the basic question clear to you? I see a bunch of unconvinced faces. So remember, what do we want to do? In my high dimensional space, let's say a person comes and gives a sample. The dot that comes from a particular sample, remember each of these dots correspond to a single sample. You want to find samples that are close to these guys because then you'll know that the phase of the sample was here. Or if that sample is somewhere here, you know that the phase is close to somewhere here. Okay. But how do you do this in high dimensions? What we are trying to do is we are trying to say that, look, we will project or reduce the dimensions to 2 dimensions, which are just the directions of largest variance. Okay? So the problem boils down to a very concrete mathematical problem. How do you find the direction of maximal variance in the data? Okay? So that's what we are going to try to do in 10 minutes. I have to teach you linear algebra. This is not going to be okay. Somehow this is okay.
Himalaya, Himalaya, Himalaya.
Okay, so how do we set up this problem mathematically? We need to formalize a little bit of notation. So what we're going to define is something called a random vector. Define a random vector, which I will call G. Okay, and my vectors, are and these are going to be column vectors. So this is G1, G2, all the way up to Gp. Okay, so this is a D cross 1 matrix. So what do I mean by a random vector? Is that each of these variables are random variables. So all that means is think about a coin toss or the toss of a die. Every time you repeat the experiment, you get different results. So what I mean by this is in each sample, if you measure the G1, you'll get different results. Okay? So I'm going to think of this d-dimensional vector as a random vector. Okay? And remember, my data matrix X is now simply, you've repeated the experiment many times and you just the G's, okay, and put them together, that gives you my data matrix. Okay, so I can think of the data matrix as simply many realizations of measuring this random vector G. Okay, all right. So the first thing that we want to define is what is called the expectation, which is nothing but the average. Okay, some of you may have seen this nomenclature. It just means that I'm taking the average of all these measurements of the genes. Okay, so this I will note by E of G, and that's simply E of G1 all the way up to E of G2. Okay, all this means, once again, if you're just thinking about it physically, is that you're making measurements of G1 many times across all these samples. You're just taking the average. Okay, that's all that this is. Just call it expectation. Okay. The important thing that now we have to define is what is called the covariance matrix. Covariance matrix of G. I'm sure all of you have heard of the word variance, right? All of you should be familiar with variance. So what is variance? If you have a bunch of data, right? Variance is you take each of the data points, subtract the mean, take the square, and add. That's your variance, right? This is a slight advance over it, where you are looking at the covariation between each of these genes. Okay? Basically, you're asking if one gene increases, whether the other gene also increases. So that's covariation. So I'll write down the definition of this. So covariance of G is simply defined, and I will call this quantity with this capital sigma. So this is simply defined as the expectation G minus G, G minus EG transpose. So see the similarity, just to show you the similarity with the standard variance that you are all familiar with. So you're taking the data, subtracting the mean. Remember, this is the mean, right? And then you're squaring it. All I've done here is I've used matrix notation. So if you just work this out, write out this quantity, you'll see that it will become the sum of squares. Okay? So then it gives you back your standard variance. So this is just the transpose of this matrix. Okay? Just write it down, you'll see that you'll get back exactly the kind of variance you're familiar with. Okay? But why did I write it like this? Because this covariance matrix then essentially becomes a D cross D matrix. Remember, D is my dimension that we are working with, the number of genes. So what you'll have is on the diagonal, you have the variances. Okay? So variance of gene 1, variance of gene 2, all the way up to variance of gene D. Okay? Remember, what is this matrix? This matrix that's telling us on the diagonals you have the variance in the measurements of each of the genes. In the off diagonals, you have the covariance, G1, G2, and so on. This is telling me what is the relationship between the different genes, right? Covariance of G1 with G1 is just my variance. Okay? So this should be okay with you. Now this matrix, it turns out, is an incredibly important matrix in physics and biology. Okay? This matrix, which is called the covariance matrix, turns out that there are incredibly interesting properties that this matrix has. So, for example, if my data matrix was generated purely from random processes, this covariance matrix has eigenvalues whose spectrum is very famous. It's called the Marchenko-Pascal distribution. And as some of you may have heard of this, it has created an entire field that's called random matrix theory. Okay? Simply by looking at the eigenvalues of this matrix, you can tell a lot about the underlying processes that are going on underneath. So in biology, in fact, this matrix is becoming more and more exciting. So for example, we have a project with a colleague of mine, who also in fact turns out to be a Stepanian, Archishman Raju, where we are looking at cancer cell proliferation, and we are showing that this matrix has an eigenvalue spectrum that has very interesting structure underneath it. Okay? So this is an aside, but I'm just trying to tell you that this matrix is an incredibly interesting matrix, okay? Both in physics and in biology. This has given rise to hundreds of textbooks on a topic called random matrix theory. Okay? Essentially, what you're trying to do is understand the underlying structure of noise in your data set. And you can do that by looking at the eigenvalues of this particular matrix. We will also be looking at the eigenvalues of the matrix, but in a slightly different context. Okay? In the context of what I told you is called principal component analysis. So where does this come up and why do eigenvalues matter? So let me. So remember what was our goal? Can someone remind me what was our mathematical goal, objective? You're trying to find something, right? Correct. And to do that, what we said is we are trying to find the direction in my d-dimensional space that maximizes the variance in the data. Yeah. Okay. So what that means is we need to first write down a mathematical expression for the variance of the data in any arbitrary direction in my d-dimensional space. Right? So let's try writing that down. That's actually quite easy to do. So what we want to now write down is an expression for the variance in my data set along an arbitrary direction V. Okay? So let me define V as a unit vector. Okay? In D dimensions, of course. Okay, remember that I have D genes, so I have a D-dimensional space. In that D-dimensional space, V is a unit vector. Okay? So if I want to now find the data along this direction, can someone tell me what I need to do? What is the simple operation that you can do to collapse this data set, my high-dimensional data set, along this direction? Sorry? Have you all heard of dot product? Yeah. Projection is essentially a dot product. You have one direction which is given by this unit vector. You take the data points which are also directions in the space, right? In a high-dimensional space, let's say this is your three-dimensional space, any data point is essentially a direction, because you can connect the origin to that data point with an arrow. So what we are going to do is, as you said, you need to take a dot product. And in matrix language, a dot product is simply V transpose G. Okay? That is the data projected along V. Okay, remember this is simply a dot. Why is this a dot product? Do you see that? Why is this a dot product? So remember, V transpose will give you something like this, right? V1, V2, Vd, right? And G is G1, G2, Gd. If you multiply this, you will get V1, G1 plus V2, G2, and so on. Yeah, that's just your dot. So I've just written this like this because I'm going to be using matrix mathematics to the rest of the thing. Okay? So this is my data projected along V. So what do I want? I want the variance along V, which is nothing but variance of this quantity V transpose G. Yeah, is that fine? We argued that this is the data projected along direction V, and we want variance of this quantity here. Okay, so let's see what this will look like. This is just a few lines of algebra. We're already out of time. Okay, I'll just take 5 minutes. Is that okay? Okay, so I'll just break that equation down into one step, and then you guys can do the rest of the algebra at home. So using the definition, remember the definition of variance comes from here. So this will give me expectation of this quantity here, V transpose G, V transpose G transpose minus expectation of V transpose G, expectation of V transpose G. Okay, all I've done is I've used that definition of the covariance matrix to write down the variation of this quantity. Okay, that's all I've done. What I need for you, this is very simple matrix algebra. You can all be able to do it. Sure. Okay. If you want, I can sit down with you all to show how this happens if you're really interested. Okay, what you'll be able to show is that this simply becomes V transpose sigma V. Okay, I'll call this equation 2. I'll call this thing equation 1. Okay, I'm happy to sit down with you guys later today or tomorrow, show you this derivation. Some of you want to do this. Okay, so this is the variance along an arbitrary direction V. Notice that this is my covariance matrix. So knowing the covariance matrix of my data, I can figure out what the variation is in any arbitrary direction. So then what is left is for us to show what V maximizes this quantity here, V transpose sigma V. Okay, that's my question, right? What is that V that maximizes my variance along this arbitrary direction? And it turns out I'm giving you the answer, but that should take maybe we wanted to show you this, that the answer to this problem is the first eigenvector of sigma. Okay? This is an incredibly powerful result, which lies at the heart of what we call principal component analysis. That the first eigenvector of the covariance matrix gives you the direction of maximum variance in the data.
Only if this is unit.
Only if this is unit. Yeah.
Divided by the.
So I'll do that. Yes. Yeah, exactly. Very good. So let's just quickly do that maximization problem. So I had talk to you a little bit about spectral decomposition, but I will not do it in the interest of time. I do recommend strongly. Have any of you heard of 3, 2, 1 round?
Yeah.
Fantastic. Have you gone through the linear algebra?
Yes.
Okay, excellent. So you know the geometric interpretation of this, right? So then you should know very nice. You should know that. So then how do you diagonal matrices are simply linear transforms represented in the eigenbasis. So you should already know this very well. So if you know this, that makes my life simple. I will just write down this maximization. Okay, so what we need is the spectral decomposition, right? So sigma can then be written down as q lambda q transpose. Remember, this is just my diagonalization formula, right? You have a matrix here. You're just representing that same linear transformation in coordinate system that is your eigenbasis. That's why you have a diagonal matrix over here. Okay? And these matrices here just comprise the eigenvectors of sigma. So this diagonal matrix will have the diagonal as the eigenvalues of sigma, right? And Q is a matrix whose columns are eigenvectors of sigma, right? So you should be familiar with this thing. So all we have to do now to find the V that maximizes this direction, as was just mentioned, is do an optimization problem, a maximization problem, which is you want to maximize this quantity here, V transpose sigma, I should be writing sigma like this, V transpose sigma V, and you normalize it by V transpose V. It doesn't matter here because this is unit vectors, but you can keep it here anyway. So here, what you will do is you say, you have V transpose, you just use this, what is called the spectral theorem. This is called the spectral theorem. You use that spectral theorem to plug in this over here, right? You have Q, the same here. So one thing, if you've not noticed, I've written a transpose here. Usually you write this as an inverse. So it turns out that, so I did not tell you this, but sigma is clearly a real symmetric matrix, right? Because the covariance matrix is symmetric. Hence, it turns out that the eigenvectors will be all orthonormal. Hence, Q is an orthogonal matrix, so Q inverse is equal to Q transpose. Okay, for those of you who are familiar with this stuff. So now you have this quantity which you want to maximize, P transpose Q transpose. I'm just writing Q transpose here because this is identity. Okay. V and this, again, I'm going to leave this to you as a simple, I mean, this is one step. So let me just write this down. Y transpose lambda Y divided by Y transpose Y, where I've defined Y to be just Q transpose V. What I've done is, remember, this is what we want to maximize. I've plugged in this quantity here instead of sigma, and then I've just defined V transpose Q or Q transpose V as this quantity Y. Okay? So now my maximization problem is here. I want to maximize this. So just one more step. Just one last line, and you have the result. Okay, so this quantity here is nothing but, you'll be able to easily convince yourself that this is simply lambda 1 y1 square plus lambda 2 y2 square, so on, plus lambda d y2 square divided by y1 square plus y2 square. Okay, and this is less than equal to lambda 1. So here, lambda 1 greater than lambda 2 greater than lambda d. Okay, so what I've done is I've ordered these eigenvalues from highest to lowest. Okay, so if this is the case, you can easily show one step that this quantity here will always be less than equal to lambda 1, and the equality will happen. Equality is happening when y is equal to E1, which is my standard basis. Okay. Why is that? When y equal to E1, your y has 1, 0, 0, 0, 0. This is my standard basis, which means here only y1 is 1. So you have lambda 1 there. Okay. All these terms become 0. Yeah. So this becomes equal to lambda 1 when y equal to E1. And notice from the definition here, when y equals to E1, what do we have? We have E1 is equal to Q transpose V. Yeah, I've just plugged in E1 over here in the definition of y. So E1 is Q transpose V, right? Which implies that V is equal to Q times E1. Okay. What is Q times E1? Remember, Q matrix has columns that are eigenvectors of sigma. When you multiply a matrix with the standard basis, it pulls out that column. So just go and take a look. So this will then be nothing but Q1. Okay, remember Q is the matrix of columns that are eigenvectors. You multiply that with the standard basis. The first standard basis, it will pull out the first column, which is nothing but Q1. Okay, so then you have maximized this quantity here when your V is equal to U1. Okay, so that's your principal component analysis. So you can show similarly that the second direction of second highest variance is going to be your next eigenvector. Okay, so then if you take the first highest eigenvector corresponding to the highest eigenvalue, you take the first two eigenvectors, you project all the data onto that. You can show that in certain cases you can very nicely order cyclic data on to these two components. Okay, people have done this, and it turns out it works well only in certain situations. But in real, it turns out for circadian clocks, this is not the best strategy. So what you have to do, in fact, is go over to neural networks and so on. But this strategy works very well in certain cases. For example, in bacteria, it works very well, where the oscillations are not that noisy. Okay, for cell cycle, it works excellently. But in human circadian clocks, you do have to add another step after this, which is neural nets. But we stop here. Okay. So we've learned something that's called principal component analysis. Okay. So we stop here. Tomorrow we'll take up the question of looking for these rhythms in single cells, and there I'll talk to you about what my lab is doing. Okay. What the future directions of this field are. Okay. So sorry, I again went over.